Okay, we're back to our cell of meiosis, and I want to remind you that you're going to use your notes that you just took in order to do this activity. And we just duplicated the chromosomes, so we're on the step 2. The spindle fibers line up homologous chromosomes at the center of the cell. So remember, your definition of a homologous pair is 2 chromosomes that have the genes for the same characteristics in the same spot. So I need to take all of my chromosomes that are the same size and shape and put them together. This is unique in meiosis. This didn't happen when we did mitosis, but all of those come together. So I just put all of my chromosomes the same size and shape together. They're now in homologous pairs. It is the spindle fibers that do that. So I want to put down some spindle fibers to remind myself that the spindle fibers are the ones that carry the chromosomes to where they need to be. Go ahead and do that step where you put all of your homologous chromosomes together and you set up your spindle fibers. Next, the chromosomes are going to line up at the center. So the spindle fibers take the chromosomes to the center. At this point in meiosis 1, chromosomes do not lose their homologous partners. So if you remember in mitosis, we were a single file. Now we're lined up and down again, but we're still with our homologous pairs. After they align, they split, and so 2 chromosomes go each way. All right, spindle fibers pull them apart. Double check that each one of your cells are going to have both the long and the short chromosomes. That's how you know you're doing this right. And meiosis 1 ends when the cell pinches together. So go ahead and do that. Meiosis 2, now we have 2 cells doing the same thing. So mine's a little bit messy here, but I have this cell and this cell both doing the same thing at the same time. I sneeze, excuse me. Sorry about that. Okay, I'm back. You can laugh at that. All right, so now I'm going to have spindle fibers that form on both sides. So I'll move one of my spindle fibers over to each side. Excuse me again. So we're going to have 2 cells now doing the same thing, and this time the sister chromatid. So each different chromosome splits. And so both cells are doing that at the same time. So you got more than one person in your group. Go ahead and help each other out. And I'm almost there. Let's see if I can fit all of my chromosomes on your screen here. The final step then is that the spindle fibers would disappear because they've done their job. And the cell membranes would pinch in, and I will have 1, 2, 3, 4 cells, all with 2 chromosomes, which is half the number of what I started with. So I just made 4 sex cells, either sperm or egg. Okay. What I want you to do now that you've seen mine is go ahead and do the whole thing again. And notice that your lab talks about crossing over. I'm going to have you do meiosis without crossing over actually while I'm gone, and then when I come back we will make sure that we talk about crossing over. So go ahead and do the whole thing one more time. Make 4 sex cells and then answer the questions. Thanks guys.